Topic 12D, epistasis. So far, we've seen that a monohybrid Mendelian cross with a completely dominant recessive allele set can give a 3-to-1 dominant to recessive phenotypic ratio, which is divisible by 4. We've also seen how two traits controlled by independently assorting genes can give a 9-to-3 to 3-to-1 phenotypic ratio, which is divisible by 16. In our next pattern of inheritance, called epistasis, we will set up a monohybrid cross and get the expected F1 dominant phenotype. But when we set up the F1 cross, the F2 generation gives a ratio of 9/16 to 7/16 instead of the expected 3-to-1. Even though we were following a single trait, this ratio is divisible by 16. So any ratio divisible by 16 suggests there are two genes contributing to this one trait. And it also tells us that the genes are independently assorting. So let's assign genotypes and try to explain this pattern. In the P0 generation, we will give each parent two sets of genes for the one trait. Each set of genes will be homozygous, dominant or homozygous recessive. In the F1 generation, all the progeny will be heterozygous for both genes and have the dominant phenotype. In the F2 generation, we can still imagine that the alleles will segregate the same way and will give a 9-to-3 to 3-to-1 ratio. But we have to interpret these genotypes differently. The 9/16 ratio, of course, is the dominant parental type, meaning it has a phenotype like one of the original parents in the P0 cross. Now the 7/16 we saw in the F2 for the recessive phenotype, we can consider it being made of 3 of the 4 phenotypic categories. So we can deduce that this 7/16 comes from combining the two 3/16 categories of the non-parental types plus the 1/16 from the parental recessive type. And this tells us that at least one dominant for each of the two genes is necessary to produce the purple pigment. And it also tells us that if one or both of the genes is homozygous recessive, no pigment will be produced. We can further explain this in terms of a metabolic pathway. Let's say that the plant petal cells make a colorless compound we'll call P1. Gene A produces an enzyme that converts compound P1 into another colorless compound called P2. Then gene B produces another enzyme that takes P2 and converts it to the purple pigment. Now, if the gene that makes enzyme A is homozygous recessive, it will fail to make an active enzyme A, so the first enzymatic step never happens, and all that's made is the colorless compound P1. So the rest of the compounds are never made, despite the fact that enzyme B is present. If the gene that makes B is homozygous recessive, then no B enzyme is made, so the second step never occurs and compound P2 is never converted. If both genes are homozygous recessive, then no enzymes will be made and compound P1 is never converted to P2. To review, epistatic inheritance is when two genes are contributing to a single trait. Ratios from epistatic inheritance are divisible by 16 if the two genes are independently assorting. Most epistatic events can be explained using a metabolic pathway model.